This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 465, recorded on October 27th, 2017. This episode is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. Get $30 off your first delivery and free shipping by going to blueapron.com/TWiV. This episode is brought to you by the Defense Threat Reduction Agency. Part of the U.S. Department of Defense, the agency's Chemical and Biological Technologies Department hosts the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Find out more at cbdstconference.com. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
Beautiful day.
Gorgeous.
Not a cloud in the sky?
That's not true. No,
there's
morning, lots of clouds in the sky.
Wow, it changed.
But it's the temperature is ideal.
I always have to guess at what's behind me, you know.
Yeah, well, I can actually look out the window.
If I look, I'll go off mic.
Uh-huh.
But when I drove in this morning, totally blue skies.
Yep.
And now it's
getting a little hazy out there.
What is it? It's 20 Celsius, maybe?
Could be.
15.
15? Wow.
A little chilly.
Well, it's about time, though, don't you think? The trees are starting to turn.
No, it's too cold. Too cold.
I like it.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
Also known as Southeastern Michigan, right?
Right. Yeah, I'm fine with that. It's 54 degrees Fahrenheit here, cloudy, and that's 12 Celsius. So I wonder if that's weather that's coming your way.
Maybe.
Yeah.
I think it is.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It is the best weather of the week today, which is fairly easy to accomplish. It's 62 Fahrenheit, 17 C, and nice. It has been sunny, but this week was just awful weather. We had a wind ripping through. We had 5 inches of rain Tuesday night. Another inch on Wednesday.
Didn't have any of that, did we? Wow.
No,
we got slammed, and apparently we're going to get slammed again this weekend on Sunday. They're talking about heavy rain and flash floods again.
Wow.
And joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Right.
We have clear blue skies and 61 degrees, which I know, Fahrenheit, which I know because Kathy is my friend, is 16 degrees C. Windy. There's a cold front coming through. This is the one that's going to catch you guys, I think.
You sound less echoey today, Rich. Did you put some...
I was going to ask if that. Yeah, put the pictures up.
That'll do it.
I got the wall, to my right shoulder is like from shoulder height to about 8 feet. I think it's a 10-foot ceiling. Is all pictures, and I got pictures in front of me and on the other side. I got dozens, and it's all family, and it goes back like 4 or 5ve generations.
Do you have a picture of Dickson there?
I'm not fan.
No, he's. I'd have to go back more generations than that. Dixon, Dixon. If we went back like 100 generations, there'd probably be some commonality.
I have a feeling you're right.
We have an in-studio guest today visiting us from right here in New York City at Rockefeller University. Theodora Hatziioannou, welcome to TWiV.
Thank you.
And I know you've been waiting patiently to be introduced, but, we have to go through the weather and everything. And you got here very early, as opposed to someone else who recently came from Rockefeller who got here, I think, after we started. I think Paul came after we started, but he probably told you to get here early, right?
Yeah, he did.
All right, we're going to talk a little bit about your recent paper. Before we do that, we're going to talk about where you're from and all of that. But first, I want to have one announcement from Basel, who wrote to us. I hope this is read before November 3rd. We get our orders, so it is, right?
Yes.
I wanted to bring your attention to this event. I know the targeted audience is Arizona-based, but since there is a webcast option, I thought I'll bring it to you and your audience attention.
Mm-hmm.
I hope we don't overwhelm the organizers. This is the Arizona State University and University of Arizona first annual joint virology symposium. And Rich, who do you think is organizing?
My guess is that Grant McFadden is in there somewhere.
Yeah, he's right at the top.
Oh,
there.
it says Grant McFadden. Yes.
Yeah. But he may have made the Eventbrite thing, but he's got his name all the way on the upper right there. Yeah.
Yeah. Well, this is the kind of thing he likes to do, and he's he's part of this Biodesign Institute.
He is the organizer. Yep. And so apparently there's a webcast option if you want to hear some of these speakers. Dixon, you're not on the program.
No, I'm not a virologist. Come on.
But the name is the Biodesign Institute. It's really
well. I was going to ask about that. What is the mission of that institute? Do you think? Is that like biomimicry, or to show similarities between biological structures, or what is it?
He is in the Biodesign Institute, Grant, and
making new life.
I don't think he has anything to do with mimicry when you're talking about like Velcro and all that stuff. But it would be interesting to know where the inspiration for biodesign came from.
Exactly.
It's from a while ago. We can ask Grant sometime, or one of the speakers here is Bert Jacobs, who has been at ASU and in the Biodesign Center since its inception. He's been at ASU forever, and the biodesign thing was initiated after he got there. And he has some fancy position in the Biodesign Institute, so he would know all about it.
Maybe it's Dickson to figure out how things are designed.
Maybe.
I found a sentence specifically to conduct use-inspired research, fuse intellectual disciplines, and value entrepreneurship.
Okay.
Wow.
Anyway,
there's a whole, and that's part of it. And then there's a mission addressing today's critical global challenges in healthcare, sustainability, and security by developing solutions inspired from natural systems and translating those solutions into commercially viable products and clinical practices.
Sounds like the lady from, where is she from?
She's in the Biomimicry Institute in Missoula.
What's her name?
Janine Benyus.
Yeah. So the idea like Velcro came from some inspiration from
the seed.
Yeah.
From a seed with a little hook on it. Actually,
what's another example of biomimicry that we use?
The gecko hand.
Would an airplane be biomimicry?
Only the honeycomb portion inside the wing. No, really, they've designed a structurally better design than the ribs that go across in the old days. This is a honeycomb design based on a honeycomb.
Right.
And
Alan,
planes do not fly the way birds fly.
No, they don't.
It turns out flapping the wings is not an effective means of propulsion.
It's not.
For using an engine.
However, if you want to fly,
but they did try that, didn't they?
Under the water,
yes, that was among the early efforts at aviation, or some of these ridiculous
kind of
things
like
that fly
how
things.
Icarus is pictured.
Correct. Yes, that's right.
Right, Theodora, you okay?
Yeah, I'm laughing too much.
You can't laugh too much.
So, wait, I wanted to also make a connection here between in the literature, the old literature of Greece.
Yes.
These fantastic epic.
You're making her laugh too much.
Epic poems. Homer used a lot of analogies to nature to describe human behavior.
Right.
And they are forever known as Homeric similes. Mm-hmm. And so one of them was Ulysses, when he was thrown out of his boat on his quest, he actually grabbed a hold of a rock and he clung to it like an octopus. Imagine that. Mm-hmm. He could never be pried loose. So that's where that comes from. So that's observing nature and then applying that to human behavior. And Homer was an excellent observer.
Yes, we Greeks were first in everything.
You were. Of course you were. Of course you were.
By studying viruses, we can learn how cells and larger organisms work.
No question.
What about that? Right?
The neurotropic viruses have opened the door for looking at the way brain behaves, right?
Yeah.
So that's I'm all for it.
Theodora, I assume you were born and lived in Greece for quite a while, right?
Yes. Until I was 18 years old.
Where in Greece were you born?
So I was born and raised in Rhodes, which is an island on the south.
Rhodes is so cool.
Are you a scholar?
Of course.
Different Rhodes. The original, not the...
What happened to that statue, by the way?
Colossus. It fell down during a huge earthquake.
Yeah, where is it, though? I mean, is the pieces of it?
The pieces apparently were sold to be made into weapons. It was made out of metal.
How could they do that? Gee whiz.
So Rhodes is an island.
It's an island in the southeast corner of the Aegean.
Okay. Is it beautiful?
Yeah.
Of course. It's the most beautiful island in the world.
One of the last strongholds of the Crusaders, right?
That's true. So it's also one of the, I don't know how many other cities have this, but we have an intact medieval city inside
Right. It's
the
amazing.
city. It is. It has the castle is preserved, the palace is preserved, everything is cobblestones or really. It's the entire thing is still standing.
Goodness.
If you've ever seen the movie The Guns of Navarone,
Oh yeah, sure.
Right.
they have the pictures of the German encampment are the trucks running all over these walls of some castle-looking thing. That was filmed on Rhodes.
Exactly.
Oh, interesting.
So it's a cool place to grow up, right?
Yes, it was great.
What's the population of Rhodes?
I have no idea nowadays.
So you stayed on Rhodes till you were 18, you said?
Well,
Yes.
It's one less than it used to be.
Yeah.
As you're here.
Yes, I'm sure a lot of people move there. So it's as far as Aegean islands go, it's considered relatively big, but you can go from one end to the other end in about 2 hours by car, so it's not that huge. And yes, so it's great growing up there. I mean, we go to the sea in the summer every day, and it was relatively, we could play outside and all the...
115,000 people as of 2011.
No?
It's pretty
Okay.
small. Wow. And you went to high school there, I guess, right?
Yes.
So were you interested in science way back then?
Good question. I don't know. So
Only you would.
I didn't know. I didn't really know what at least biological sciences were. So in Greece, the system is that you basically have to decide what you're going to do for the rest of your life, because you decide where you're going to go to university and then what career path you're going to follow. The university is very rigid, so you don't get room to maneuver once you're in. To decide which avenue you're going to take, and you at the end of 18 years old, you take this countrywide university entry exams, and they're essentially split into categories, and you can either choose science or classical studies, essentially. And I was actually quite good at both. So I had these two choices. And on the one hand, I thought if I take classical studies, what I would like to do is become an archaeologist, which is a great profession in Greece, right?
Yeah.
Sure.
But if I did that, I could see my career and life unfolding in front of my eyes. I knew exactly what I was going to do. The other option would be to do science. And I liked math, and chemistry was not really my favorite subject. But I had heard about this new area, at least in Greece it was new, called biochemistry. And you couldn't really specialize in biochemistry in my country when I went to university. They had a 6-month course in one of the universities, and that was it. So I thought, okay, I will go somewhere else where I can do this new thing, biochemistry. And at the time, Greece had joined the EU. So I was, tuition was essentially greatly reduced if you wanted to study abroad. And the British universities accepted our grades for entry. So I went to Bristol in the University of Bristol to do biochemistry.
Lovely.
That's where you got an English accent, is that right?
Yes.
Which is fine, right? But did you know English?
I spoke English very well before I left. Most Greeks speak English quite well.
Okay. And after Bristol, where did you go?
So after Bristol, I went to Imperial College to do a master's. So I did not. So perhaps not unsurprisingly, I found the first year at Bristol very hard because kids that grew up in Britain and did the A-levels, they already had some experience in practical. We had zero. We had an old lab in my high school that nobody ever used. So I'd never done an experiment. They had some flasks and some weird looking things, but nobody did anything with them. So I still remember my first class in physical chemistry. I was completely overwhelmed because I did not understand anything. And later I found out that my other fellow students also didn't understand anything, but didn't show it.
Yeah,
they were better at faking.
Yes, exactly. But the university was very good, and they allowed me to take other courses that will help me build up my practical expertise. And but still, I mean, it was the first time I was away from home, and I didn't study that hard. And I found that being in the lab was interesting, but the way they taught biology or biochemistry was very dry. So showing me over and over the energy cycle in the membrane of mitochondria year after year, and I still don't remember it. It's just didn't sink in. So then after I finished, I thought maybe I want to do a PhD. I wasn't sure. I certainly didn't have the grades to go straight into a PhD program, and I thought I better learn something now. So I went to a biotechnology master's programs at Imperial College London. And there they allowed you to spend 3.5 months in a lab actually doing projects. So I did that at the MRC, was actually trying to crystallize a protein. I don't remember what protein, but I expressed it in bacteria and set up this crystallization hanging drop trays. And I found that quite interesting and more engaging than the mitochondrial energy cycle.
Cool. After that, you went where for PhD?
So after that, I realized that I still didn't have a full grasp of what it's like to work in a lab. And I thought that if this is what I want to do for the rest of my life, I want to know what it's like to do it for an extended period of time. So I stayed in London for a few months after I finished my master's looking for a job. And I was lucky enough to get a technician position at the lab of Robin Weiss. He was at the time at the Cancer Research Institute in London.
Right.
That was still at Lincoln's Inn Fields?
No. It was near Imperial College, actually.
Okay.
So it was Kensington.
Okay.
Oh, it's a lovely neighborhood.
Yeah, it's beautiful.
Good shopping.
I didn't have any money to shop at the time.
Isn't Harrods right down the block?
It's not too far.
That's right.
Yes. I remember passing by.
So that was probably where you first encountered retroviruses?
Yes, absolutely. So I was working, so Robin was already very well established. Of course, I knew nothing of that at the time. But he had multiple PI, like subdivisions working in the lab for different things. And I worked with Thomas Schultz, and we were working on HTLV1 envelope. At the time, the receptor wasn't known, so we were hoping maybe we'll find the receptor. But my main project was to try and produce and purify the envelope protein. But then the Kaposi sarcoma virus came along. So there was a big effort in the lab to try and find it. They didn't find it. It was a different group that identified the virus. But we had a lot of things set up. And of course, Kaposi sarcoma is endemic in Greece. So I helped Thomas get in touch with the Greek physicians, and they sent us samples, and I did a lot of sequencing.
Nice.
And I also tried to develop a peptide detection assay before I left. And those actually are papers that I'm on that are very, very widely cited because they were the first papers on human herpes virus 8.
So it was discovered here at Columbia. Chang and Moore were here at the time.
That's right.
Later they moved to Pittsburgh. But I remember that they used to come and talk to Saul all the time, who had been in herpes virus his whole career, and slowly they teased this out. It was very interesting. Yeah. So that sounded like a great experience in Robin's lab, right?
It was.
And did you decide you liked lab work at that point?
I fell in love. It was great. I loved working at the bench. It was obviously a phenomenal lab to be in, and the people that were there, and it was just amazing.
So we often get a lot of emails from people who want to know, how do I get into this field? How do I decide if I like it? Would you say working as a tech is a great way?
Absolutely.
Yeah.
I think that working as a tech gives you a real feeling of what it's like to do this as a job. And as a job, I always, whether you really love it or not, you will know straight away.
I'm the senior member of this group here, and that's exactly how I started also. No kidding. In 1962, I was a technician here, and that just I got hooked.
Yes, exactly.
Yeah.
That's
the
it.
thrill of discovery is just, you come in the next day and nothing happened, and all of a sudden something happened.
Yeah.
And you get to see it, and that's great.
Where did you decide to go to graduate school then?
So, being in Robin's lab, I obviously interacted with a lot of people, and so I had choices. And I had at the time decided, I need to get a PhD if I'm going to stay in this career. And so I had the opportunity to go with Thomas. He was moving to get his own position at Liverpool. But I had also met, there was this young French PI working there at the time, François Cosset, who was also going back to France, Lyon, to start his lab. And he asked me if I wanted to join him and get my PhD there. And I said, sure.
It's a nice thing about Europe. You can easily move to different countries.
Yeah.
Right?
Yes.
Whereas here, we can move to a different state.
It's true.
But different states here are almost like different countries, no?
It can be. They can be.
I can attest to that.
Oh, yes.
Exactly.
Oh, yes. Now, what did you work on there in Lyon?
In Lyon, we worked on trying to use retroviruses as tools for gene therapy. So essentially trying to, actually it's almost what I do now, expand their tropism, target their retroviral particles to specific cells by manipulating the envelope, either by using envelopes from other viruses like influenza, or by adding things onto the retroviral envelope and trying to target it to specific cells.
By the way, Robin Weiss, we have to get on TWiV at some point. Yeah, that was his own episode, because he's one of these people in the field so long, has done so much. So interesting. All right, what was next?
So next was America. So everybody in Europe said that if you want to do a postdoc, you have to go to America. It's just a different experience. And while I was a PhD student in France, I attended Cold Spring Harbor meeting, and that's where I met Paul. And
he told us that. Yeah.
So we decided that he was already in America. So I thought, okay, I want to join you, and we're going to try and move into the same city. And we ended up in New York. He started his lab, and I actually came next door to Steve Goff lab.
That's right. That was a postdoc, right?
As a postdoc, yes.
I used to see you here. That's right. Running.
Yes.
But you were running around so fast I couldn't quite tell who you were. But I remember. How long were you here?
About 2 years, a bit over 2 years.
And you continued to work on retroviruses, obviously.
Yeah,
right?
So it's like many scientists. I had read a review that Steve had written on FV1, and I wrote to him and I said, I find this subject very interesting. I'd like to come over and work on this. And he told me, well, I already have a person working on this, but I have these other resistant cell lines. Why don't you have a look at those? So I worked on those with Gungxia Gao. He was a postdoc, more senior postdoc in Steve's lab. And that was also great. It was very nice working with Gao on this project.
Okay, so that was your American thing, but then you're still here, so I guess.
Yeah, well, it's
that's to stay.
Yes, it's still a great country to do science in.
Yeah. When you first came, were you thinking of eventually going back to Europe?
No.
Not necessarily.
No.
Because you had left Greece years ago, and
Yes.
there's no pull anymore, right?
No.
Yeah.
But when I left Greece and after a couple of years abroad, I knew I was not going back. I did explore it briefly at some point, but it was just
Was Arj Stratiatus here while you were here?
No, he wasn't.
No.
Because when Steve moved to this floor, that's because Arj had been here, and he left.
See. You would have loved meeting him. By the way, he was.
Oh yeah, Alan, you remember Arj?
Oh boy, do I remember Arj.
He was fantastic.
He was a very He was from Greece, also. He was extremely animated, opinionated.
Yes.
He
That's
would
right.
be yelling at people all the time. You could hear him through the walls.
Well, the three offices, Vincent's office was right between Arj's office and Saul Silverstein's office. And it was hilarious, just the interaction. Because Vincent, was a pretty quiet guy sitting there in his office, and there'd be these loud arguments going on both sides of him all the time. It was very fun.
Yep.
I like he was just a minute diversion. He wore black every day. Black shirt,
yes.
Black pants every day. And when he came in, he'd have one of those Greek sailor hat type things.
Yeah.
He'd wear it, take it off. He'd smoke all the time. Back then, you could smoke in your office.
Yes.
Yeah.
He was a really interesting guy, but he had this.
He was eccentric.
Never yelled at me, but he would yell at people. Yep. And Saturday morning, if you weren't there, he'd call you up. Where are you? Why aren't you here? Yeah, for sure. And then he went back to Greece to be director of something. I don't know what.
Right.
Really interesting guy. And then after your postdoc here at Steve, I kind of lost track of you. Where did you go after that?
I went to work with Paul, yeah.
At the Diamond?
Yes.
That was on First Avenue all the way down there, right?
First and 28th Street. So that's opposite NYU. Or actually Bellevue.
Okay.
So, yeah, Paul had his lab, and it was, he was young and unknown, and he had a hard time getting postdocs to apply. And also we were thinking down the line, if we have a family, I don't really want to, it would be better to be at the same place. It would make things easier. So, and we talked about science at home all the time anyway. So
yeah, it's nice to be able to talk about it all the time. Well, we do that on podcasts now. Right?
Yeah.
And so then at some point you moved to Rockefeller from the Diamond, because the Diamond
that's very recent. So I stayed as I was a postdoc in Paul's lab for a few years, and then gradually got my own group.
Yep.
And then I just earlier this year in January, we moved to Rockefeller.
Oh, so you're a newcomer.
Yes, very new. That's why I don't know anybody.
I'm trying to find that picture. You were in that picture with famous people that he showed us last time, right?
With the Bill Clinton.
There it is. Man,
Yeah.
I just searched for B. Nash Theodora and I got it. And there you are. And Bill Clinton, yeah. And David Ho
Yes.
also.
There's another one with Magic Johnson.
Yes.
I'm not in that one, I think.
that's
Paul
right, you're
is
not
in
in
that
that.
one. But I think I also have one with Lucy Liu.
Yes.
He did a...
Whoa, look at this one. This one has Lucy Liu, Magic Johnson. You're not in this one. Bill Clinton, David Ho, David Baltimore.
Wow.
But then there are others. There's so many.
What was that event?
So that was a gala that was organized to raise money for Aaron Diamond. It
Goodness.
was, and they gave the Irene Diamond Award actually to Bill Clinton for his contribution to help.
Yeah, here's one with you and Lucy Liu.
Mhm.
Did you get to talk to her at all?
Very little.
Very little. And then Beatrice Hahn,
Han, yes.
who would be more interesting to talk to, right? And then David Ho. Although, who knows? Actors can be interesting, I guess. So why did you and Paul and others leave the Diamond? Was it the building was taken over or something? Or?
It's part of it was that the Diamond is about to change anyway, and then Paul had already gotten his tenure at Rockefeller, and we decided that if Aaron Diamond was not going to join Rockefeller, then we would rather stay at Rockefeller. So our labs moved.
So you're no longer part of the
No,
Diamond
we're
at
not.
all? No,
Not at all.
And the Diamond is not at First Avenue anymore. Is that correct? Or
It
still
is at the moment.
Oh, it is.
But it might come here.
Oh, Columbia. That's right.
Yes.
Yeah. I've heard that. I've heard that rumor. Yeah.
So I guess it wasn't forever.
Diamonds, yeah.
No.
Just adding a new facet to their
It's not that...
talents.
It wasn't that kind of diamond. Anyway, because it's funny because I got this question when I went through immigration one year. The person asked me, where do you work? And
Diamond,
I said, on Diamond. And they said, oh, do you work on Diamond?
Yeah,
and I
no.
said, no, I don't.
It's funny.
Tell him you study baseball.
I knew
The Diamond District.
I knew an investigator at Wayne State. Her name was Faye Right Hand.
Oh,
Oh my God.
And she went to order a car once, and they made it with the steering wheel on the right hand side. Can you imagine here in the U.S.? She said, well, we thought you wanted on the right.
Wow.
This episode is brought to you by Blue Apron. Blue Apron's mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting the highest standards of ingredients, and building a community of home chefs. For less than $10 per person per meal, Blue Apron delivers seasonal recipes along with pre-portioned ingredients so you can make delicious home-cooked meals in 40 minutes or less. You can customize your recipes every week based on your dietary preferences, and there's no weekly commitment, so you get deliveries when you want them. They have a freshness guarantee, which promises every ingredient in your delivery arrives ready to cook, or they'll make it right by shipping the exact amount of each ingredient for each recipe. Blue Apron is reducing food waste. This October, Blue Apron is celebrating its 5th anniversary by bringing back its top 20 recipes as picked by the Blue Apron community. And this is for a limited time only, so check it out. Blue Apron is all about giving people fresh recipes to explore as they learn to cook new dish after new dish, which is why they don't normally repeat recipes within one calendar year. That's what makes this limited time offer so exciting. Now, I've tried Blue Apron, of course. I'm not a fan of shopping. I don't like going to supermarkets. I always forget something. So this is great. Blue Apron, the box comes right to the door. It's everything you need. It's all measured out for you. Easy to do. There's a lovely recipe card. You can get almost anyone interested in cooking. You say, hey, let's do this meal in 30 to 40 minutes. Here's the directions. Let's do this. Really easy to do, and it'll be delicious. And the meals that we have made are great. They're just wonderful. I absolutely love it. And for people that are busy or just want to learn how to cook, or even people who know how to cook really well, it's great to try different things. So check it out. Try Blue Apron's all-time consumer favorites. Go to blueapron.com/TWiV to get $30 off your first delivery and free shipping. That's blueapron.com/TWiV. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron, so don't wait. Blue Apron's a better way to cook. This episode is brought to you by the Defense Threat Reduction Agency and the meeting that they're sponsoring. Imagine an everyday, inexpensive drone you could buy online modified by terrorists to spread chemical or biological weapons over a crowded football stadium or holiday parade. Plague, VX, sarin, flu. How could we prevent a scenario like this from happening? How could we counter the effects? How could we treat the victims? Join us in Long Beach, California, November 28th through 30th for the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Collaborate with more than 1,500 scientists, subject matter experts, military science members, industry partners, and academic leaders from across the globe who are committed to making the world safer by confronting chem-bio defense challenges. Part of the U.S. Department of Defense and charged with safeguarding our warfighters and our nation, the Defense Threat Reduction Agency's Chemical and Biological Technologies Department hosts this important conference and brings together the best and brightest from around the world. Please join us to share your important ideas. For more information and to register, you can go to cbdsdconference.com. You can follow the progress of the meeting, who's speaking, and so forth at Facebook. Search for DODDTRA. And on Twitter, it's CBDSD Conference. The 2017 CBDSD Conference, today's innovation, tomorrow's warfighter capabilities. Most advertisers ask us to talk about your experience with the product. I don't think I can do that.
No. I'm not dead yet.
That's right.
yes.
Now, you've just published a paper in Nature Communications, and we thought we would talk about that. And its ApoBec C3H structure reveals an unusual mechanism of interaction with duplex RNA. Authors are Bone, Thumar, York, Raymond, Brown, Benash, Hatziioannou, and Smith. And this deals with ApoBec C3, of course, but I wanted to first back up to 2014 to your science paper. And this is what you talked about at ASV, mainly. HIV-1 induced AIDS in monkeys. This was about a pigtail macaque model that you developed.
That's right.
So at that point, were there any models at all that you could use to study HIV-1?
Of course there were, but they don't use HIV-based viruses. So because HIV-1 tropism is so limited to humans, so it's become extremely specialized for its host, it doesn't replicate or cause disease in other species. So what you can use is surrogate models, infect monkeys with simian immunodeficiency viruses, but they are genetically quite distinct from HIV-1. So all these models have limitations. You can still do a lot of studies with them, but it's far from a desirable model. For example, if you develop let's say, vaccine using antigens against HIV-1, they will not limit HIV replication.
Why did you decide on pigtail macaques?
That's a long story. Because that's what basic science told me to do. So this is a project I started when I was still a postdoc with Paul, and we obviously work very closely together on this. And it was to study what we call now restriction factors. And when I first started my postdoc with Paul, this field had not exploded like it has now. And one of my first papers in Paul's lab showed that you have this specific species block of lentivirus infections if you test cells from different species. And we showed that HIV-1 could not even infect rhesus macaque cells. And then we were looking for the factor, but the Sodorsky lab found it first. So it was TRIM5 alpha, so very, very potent inhibitor. And subsequently, ApoBec3 proteins came, and then tetherine and MX2 and some others. So we realized that at least in rhesus macaques, TRIM5 alpha and ApoBec3 proteins were very, very difficult to overcome. So we tried to manipulate HIV just by adding pieces from SIV that could help it overcome these proteins. It didn't work for rhesus, and I suspect it's because the capsid plays such a fundamental role in so many different events in the virus life cycle that making chimeras with capsid is just bound to have some small defect that eventually will kill the virus. So, but we was, as part of our studies to look for restriction factors, we were characterizing restriction factors in other monkey species. And we actually 4 other labs at the same time published, there are 5 papers identifying that pigtail macaques don't have a TRIM5 protein that recognizes HIV-1 capsid. They express a different protein called TRIM-CIP that actually arises from retrotransposition. That's actually very interesting. But the TRIM-CIP fusion protein that's present in pigtail macaques just doesn't interact with the HIV-1 capsid. So essentially, for what we want to do, pigtail macaques are TRIM-5 null.
So this was all done in cells,
yes.
Okay, so you got some.
And did you figure that out just by screening a bunch of different cells?
They're not a bunch of different cells, actually. There are a few cell lines, and you basically PCR out the gene of interest from all these different cell lines and characterize what they express. So we were trying to PCR up a TRIM5 alpha from pigtail macaques, and we're failing. And in the end, TRIM-CIP was discovered, and we thought, all right, let's try TRIM-CIP. And then we thought, oh, that's great because we know how to manipulate the capsid cyclophil interaction. But it ended up we didn't even have to do that because it's TRIM-CIP protein in pigtails is just does not see HIV-1 capsid.
So you can infect pigtail macaque cells and culture with HIV-1.
Yes, you can.
And it will be
Single round.
single round makes virus particles.
It does, but then they are crippled because of the picton macaque Appleback 3 protein.
Right.
So the picton macaque Appleback 3 proteins are active, but that's okay because the junction, it's easier. Well, at least it was easier to manipulate HIV to replace its VIF. So viruses like HIV-1 counteract Appleback 3 proteins by expressing a small viral protein called VIF that essentially targets Appleback 3 proteins for degradation, reduces their concentration in the cytoplasm so they don't infiltrate the budding particles. Because Appleback 3 proteins, they essentially hitch right into viral particles and then deaminate the single-stranded DNA intermediate during reverse transcription in the subsequent replication cycle.
Weapons of mass deamination.
Exactly.
WMD. Remember those? Okay. So you
We
figured
looked for them but couldn't find them.
so you figured you would be able
We did. I'm sorry. Appleback 3 are real.
See?
Yes, that's true. So you
Stealth.
figured
We're all
you
stealth.
could easily overcome the Appleback problem in pigtail
So we
cells, right?
yeah. So with this, if we manipulate, we're able to manipulate HIV, replace the VIF with the VIF from SIV-MAC. So the HIV-1 VIF targets human Appleback 3 proteins, but doesn't target macaque Appleback 3 proteins.
Right.
But if you use the protein from SIV-MAC,
Right.
which is a virus that replicates in macaques, then obviously it can counteract them.
All right. So that gave you courage to actually do an animal experiment.
Yes. So we had the results in vitro, and you could you could infect, as you said, you could infect the cells with HIV, but it didn't replicate. But with our virus, it replicated.
Yeah. With the SIV VIF, it would replicate.
Yes, it would replicate. So it told you that Appleback 3 overcoming Appleback 3 proteins was
the
necessary and sufficient, to get replication.
so now you have a slightly modified HIV that replicates in macaque cells.
Exactly. So it's only 6% of the genome that's not HIV-1, which is a great improvement over previous models.
Okay.
So, yes. And then, as you said, so now what do we do? So we're fortunate to know very well researcher at NCI Frederick, Vineet Kewal-Ramani, who told me, let me talk to Jeff Lifson, who is also at NCI, about this and see if he's interested in testing your virus. And it turns out that Jeff was very enthusiastic. He was actually great. And he gave me the animals and said, look, I'll put in the animals and you can send me your virus and we will do an infection. And
he gave you like a FedEx box, showed up full of monkeys one day?
No. They came in my apartment.
They
I
sent
didn't know
them,
what to do with them.
Alan, they sent the barrel of monkeys.
A barrel.
Yes, a barrel. No, I mean, he was willing to.
He was willing to do
the experiment for me. So, and I didn't have to pay for the monkeys. That's what I meant.
Yes.
Because of course at the time I had no funding for this project. It was too.
Neither did you have monkeys, right?
No, and no experience working with animals. So this was a completely different beast for me. So he did those experiments, and it turned out that the virus replicated in the monkeys exactly like it replicated in tissue culture, which was amazing. And that was our first paper. And then, of course, that was a replication that it was eventually controlled in the animals. So we tried to make it better by serial animal-to-animal adaptation. And
gain of function.
Yes.
Exactly. And we did that. And that's what the 2014 paper shows, that eventually we obtained blood from an animal that when you use it to inoculate other animals, they progress to AIDS-like disease. It's exactly the same symptoms as what humans used to die from when the HIV first came around.
So if you infect enough monkeys with enough viruses.
Passaging is the key here. Yes. So you have to adapt. So the first 3 passages, we could see gradual improvement in viral loads, gradual CD4 depletion, which is a hallmark of HIV-1 infection, particularly during the acute phase, but nothing else. Okay. But then passage 4, 1 out of 3 animals actually progressed to AIDS. And after that, subsequent passages had the same result.
So that's
And
interesting. That it gave the same pathological endpoint that HIV in humans reached.
Yes, but that is why macaques are such a useful model for studying HIV-1, because when you do infection with these viruses, and even HIV-1 related viruses, the symptoms of the disease match what humans progress to very closely. So that is why it's the preferred model.
Right.
What's the difference between that evolved pathogenic virus and the starting material?
Multiple differences, and we haven't finished analyzing them all yet. One of the biggest changes was the acquisition of the ability to overcome macaque tethering. So when we first started our passage in the animals, we didn't know about tethering. Paul and John Gotelli found that restriction factor a few years later. And so it was very, very gratifying to see that one of the first adaptive changes that occurred in a virus from passage 1 was an insertion in the transmembrane domain of VPU, which is another small viral protein that antagonizes tethering, that allowed it to antagonize macaque tethering specifically.
Tell Dickson what tethering does to HIV.
Oh, tethering.
All the other non-virologists listening.
Yes. Tethering is perhaps the only appropriately named restriction factor. It does what its name suggests. It physically tethers viral particles on the surface of the infected cells. It traps them. It physically traps them so they cannot go out and infect other cells.
How does VPU antagonize this?
So the transmembrane domain of VPU interacts with the transmembrane domain of tethering in a species-specific manner, and then removes it, traps it before it gets to the plasma surface, plasma membrane, or also removes it from sites where particles assemble. So.
That's very cool. But you think there are other changes also?
Oh, there are. So we have some more recent changes that suggest that maybe there's some, at least partial adaptation to MX2. And another recent paper that we had shows that the envelope has adapted to use macaque CD4. Mm-hmm. So this is another area that has recently become, at least people in our field have become aware of, is that macaque CD4 doesn't function quite as well as human CD4 as the primary receptor for HIV-1. And our virus envelope adapted to use macaque CD4 better.
I noticed one of your animals also developed pneumocystis pneumonia.
Yes.
What happens in people, right? That was one of the earliest defining infections, opportunistic infections.
Exactly.
And that presumably just acquired that, or maybe they're colonized with pneumocystis already, and it just got out of control.
Yes, I think it's just a question of the immune system not being able to control the secondary infections.
I wanted to ask you about one aspect of the macaque model. That is, in the beginning, you had to deplete CD8 cells in order to get replication. Is that right?
So we still do. We need to transiently deplete CD8 cells at the time of infection to have progression to AIDS. So this is achieved by injecting an anti-CD8 antibody in the animals that depletes CD8 positive cells from the periphery mainly, somewhat from the lymph nodes. It has no effect on the gut. So, and it's transient. It lasts for a little while. But if we do 1 or 2 weeks, and then the CD8s come back. So if we do that at the time we administer the virus, the animals progress to AIDS. If we don't do that, we get really high acute viremia. We get some CD4 depletion, particularly in the gut, but the animals manage to control virus replication.
Interesting.
And it's important that you do it at the time of inoculation. If we do it later, we have rebound viremia, but it's transient. It goes, it's controlled as soon as the CD8 cells come back.
So even with the final virus after this multiple passages, which you now use for all your experiments, you still have to deplete the CD8 with that as well?
We do. So what we are trying to do now is to adapt the virus to replicate in non-depleted animals. So again, by serial passage to obtain a variant that doesn't require this manipulation. Should have additional mutations aside from the ones you talked about here, right? Yeah.
Now, that's interesting because I met someone at, I think it was Wisconsin, who was very interested in the role of CD8s AIDS patients, right? Because he says it's pretty much ignored, but he thinks they play an important. The ratio of 4 to 8 is really important.
I wouldn't say ignored. The HIV field seems to go through phases. So there's a few years where everybody's saying, no, it's antibodies that do everything. And then there's years after that, people say, no, it's CD8s. And then they go full cycle again and again. I think all parts of the immune system play a role.
Yeah.
It's interesting in our model is that it's also kind of puzzling. We still haven't figured out what the exact mechanisms of CD8 cells being there control not the acute outcome of the outcome of the infection during the acute phase, but what happens eventually. And now that's we haven't managed to nail that down yet.
So in your model, you deplete the CD8s and you infect. Now, as the infection proceeds, do the CD8s come back?
Oh, yes, they do. So it's as I said, it's a transient depletion. So you inject the CD8 antibody at the time you administer the virus, and within a week or two, the CD8s come back. But it's too late. The virus has already taken over, and the animals progress to AIDS.
Got it. So have others now gotten your virus and used it in macaque studies?
Not yet, because we haven't developed an IMC yet. So sorry. We have an infectious molecular clone of the virus. So monkey blood is not generally abundant.
Yes.
So, but we are about to get that.
That's an interesting acronym, IMC.
Yes.
I want to go back to the tethering for a moment.
Sure.
So this is a living cell. The viruses are set to bud. Instead, they're intercepted by the tetherin, and then they're kept in place, and they can't escape, so therefore they can't infect another cell. What happens when this cell dies? Are the viruses still infectious? What happens to the virus after it's tethered?
I guess it just gets back in.
Is it then digested by a lysosomal-like vessel or something?
Eventually, yeah.
Yeah.
Okay.
I believe that's what happens,
fine.
It's digested.
Check it out.
It goes back in whence it came.
There you go.
Do you like that?
I do.
Whence.
And
so you continue to, now you have your own macaques, pigtail macaques, I presume. You don't have to get them from NIH anymore, right?
No. I'm still working very closely with Jeff, and because it's, as I said, he's a great collaborator. I couldn't have asked for a better person to work with. And he has assembled a team of people that are really experts in what they do. So there's Greg Del Prete and Brandon Keel and Jake Estes, who each specialize on some aspect of our infected animals. And it's been absolutely amazing working with them. And Vinit and Paul. We just had a teleconference yesterday, and we decide what to do. Yes, I know. I send you the email by accident. But
so one of these co-authors has a V in this. Yeah, Vinit. So she types Vinit and my name pops up.
Yes. And of course, I only recently started corresponding to you more frequently. So before it was only one Vin. So
now
never checked it. Now
it's dangerous. Yeah. Well, fortunately, I understood it wasn't meant for me. So you do all the animal experiments in collaboration with them. There are no monkeys in New York. No pigtail macaques in New York.
No.
Not even at the Bronx Zoo?
That's a joke. Trying to make a joke. There are no pigtail macaques in New York. You don't get it? Okay. Well, maybe there are at the zoo.
I don't think.
Might be.
You don't think so at the zoo? No?
I've been to the Bronx Zoo. No.
But
are Japanese macaques at the Central Park Zoo. But they're different.
Those are the ones that hang out in the thermal springs.
Exactly.
Some beautiful pictures of those animals.
Dickson asked you before we started about where pigtail macaques are from.
Right.
So they're Asian macaques also. Okay. Part of their region where they live overlaps with rhesus macaques. So rhesus are in India and China, and then pigtail macaques are more southern China, Indonesia, and further south.
So in your paper in the discussion, you say, our findings suggest that evasion or antagonism of antiretroviral proteins is key for colonizing new hosts, right?
Yes.
But you say antiretroviral protein. You don't say restriction factors.
Paul doesn't like restriction factors.
Nah. How about you? Do you like?
I like it.
You do?
We don't agree on everything.
Well,
Thank goodness.
it's a uniquely retroviral word, right?
Yes.
And other viruses have their own. Like we call them interferon stimulated genes because many of them are just ISGs, right? And many of them there's over 1,000, right? And they all have specificity for different viruses, and so we don't call them restriction. So somehow this got to be something the retrovirus field used.
Yes, because they're not, as you said, yes, a lot of them are induced by interferon, but not all.
Not all,
and
yeah.
some of them have a quite high basal expression level. So you can you can amplify TRIM5 and ApoBec3 from cells without having to treat them for interferon. They are expressed. They're already there. Yeah, interferon produces more, but that's why I like restriction factors.
They remind me of restriction enzymes.
Yes, so it's the same thing.
Yeah, right? Because that restricts foreign DNA in bacteria, right? So it's okay. Well, you will see when you come. Can I tell people that you're going to come?
Oh, sure.
So, Theodora is going to join Principles of Virology team. Because Ann Skalka is phasing out. Well, as a writer anyway, she's going to be slowly leaving, and we asked Theodora to join us, and we're happy that she did. And
you'll get to have lots of discussions about word choice.
Yes, you will see.
Yes,
I think there are people that don't like restriction factors, Anne in particular. But I'll be amused by your discussions. That'll be fun. We're having our first meeting next week.
Yes.
So let me ask you something that's you may not know the answer to, though you possibly will. So given that last statement, is it also true that the adaptation of SIV from chimpanzees to humans, in the early 20s to make HIV, did that also involve overcoming?
Absolutely.
As you say, restriction factors?
Yes, it did. So one prime example is actually tethering.
Mm-hmm.
So the SIV that infects chimpanzees and most SIVs, so VPU is a protein that doesn't exist in all lentiviruses. So most of the lentiviruses that don't have VPU use a different viral protein to antagonize tethering, and that is NEF. So the SIV that infects chimpanzees uses NEF to antagonize the chimpanzee tethering. Mm-hmm. And now tethering is one of the few restriction factors that differs between humans and chimpanzees. So the human tethering, so NEF targets a different part. It targets the cytoplasmic tail of tethering. So the human tethering lacks 5 amino acids that are the target for NEF proteins. Mm-hmm. So the chimpanzee virus could not use NEF to antagonize human tethering.
Right.
But it did have VPU. So the chimpanzee viruses that change their VPU to antagonize human tethering are the ones that are responsible for actually the group M, which is the main group of HIV-1. Yep. So here's another example where adaptation to the species-specific restriction factors was crucial for the virus to adapt to a new host. And
does this apply to HIV-2 as well?
HIV-2, I don't know.
Okay.
We haven't looked at that as much.
It's very neat. Mm-hmm. I mean, it may be the case that this is the best understood species jump, right, of all the viruses that go from one to another.
Yeah.
So, I mean, there's a fundamental question that this raises, though, of course, because these restriction factors were not there for just restricting viruses. They were there for a biological purpose.
That is the biological purpose.
Well,
the biological purpose is restrict viruses.
So Richard actually asked the question I was going to ask them that. What was their day job? So, what did these proteins do when they're not restricting viruses? Do they do some function for cells also?
They wait to restrict viruses.
Okay.
Exactly.
All right.
What you say is true for virus receptors. They're probably there for other reasons, not to bring in viruses into the cell.
Yeah.
But these restriction factors are probably the defense threat reduction agency of the cell.
Oh, great.
Excellent.
There we have a tip to our sponsor. Yeah. Very nice.
Perfect.
That's really good. I like that. Well,
that's cool. That's great.
Now the paper that we have you here for. You thought it was for this paper, but
so wait a minute. As long as we're on that topic.
Oh, you're still there.
Yeah, I'm still here. So
pursue it.
If ApoBecs, when we talk about ApoBec, we talk about other restriction factors as well. But let's focus on ApoBec. If its sole purpose is virus restriction, does it work on viruses other than HIV?
Some apobecs do, yes.
Okay.
Some do.
But
By a
apobec-
similar mechanism?
Yeah, I don't remember right now, but I
Okay.
think so.
Okay.
don't apobecs have some role in cancers also?
It has been, yes, that has been proposed. It's and
Okay.
I think the data is there's conflicting data in the literature at the moment about that. That's apobec 3B that
Yeah.
doesn't actually have antiviral activity as far as we know, at least in humans.
You can imagine that. I mean, maybe of all the restriction factors, maybe this one as a deaminase could have some other function, right?
Yes, absolutely. But most of these are in the cytoplasm, so they target DNA. So
Most
What
of the,
about,
Go ahead.
I was just going to say things like apobec 1, at least what I found here just now says it's linked with cholesterol control, cancer development, and inhibition of viral replication. So it's a different, I mean, it's not apobec 3, but
yes, I have to confess ignorance on apobec.
I was wondering about that because I think it's in the paper that we're going to discuss today, or maybe something I looked up. The apo part of it comes from apolipoprotein.
Yes.
Correct.
Right.
Okay,
That's
so
true.
It has a lot of other functions.
yeah.
Yes.
And so the full name is apolipoprotein B mRNA editing enzyme catalytic polypeptide.
And in fact, some
And then it can be 1 or
apolipoproteins-
3 or whatever.
yeah,
Like,
just-
I think there's a like in there somewhere.
Polypeptide-like.
Yes.
some apolipoproteins.
just
Okay.
rolls right
Not
off
in
the
this-
tongue.
Yeah, and it doesn't mean anything.
Well,
Yeah,
and the
well,
other thing is we're
that's why I was
talking about
wondering.
other functions of these proteins, not all of which we necessarily know.
Yeah, right.
Exactly.
But the known functions for a lot of these are simply virus inhibition, which could be enough.
Could be.
It
So
would be enough to keep it there, sure.
what happens in a cell culture if you knocked it out? What would it result in, do you think? Without viruses, just to see if it had cell function also.
But so I don't think that a cell culture experiment is the appropriate one because look at them during species evolution. Particularly for apobec 3, the locus has expanded during evolution, and in primates we have 7 of them.
Right.
So, and they target viruses in slightly different way. So clearly, it was done in a way to overcome new infections and adaptation.
And some of them function to restrict infection with trypanosomes as well.
ApoL1, I think.
ApoL1,
Yeah.
that's right.
So I think Harmeet Malik has studied the expansion in response to what we think are virus incursions, right? So apobec C3 is the one that HIV.
Not apobec 3, not apobec C.
Yeah, that C
Apobec-
fools me in there. Yeah.
Apobec 3.
Apobec is already part of the back.
Exactly.
Apobec 3 is the one that HIV worries about.
Yes.
Not others,
Yes.
right?
Not others.
Okay. Tell us a little bit of the story of this paper then.
So how this paper came about is, I don't want to call it an accident, but
Science is like that.
yes, it is. It is serendipity.
Can be, yeah.
Isn't that an island off the Greek coast of...
No, that's Santorini.
The role of Santorini in science.
Yes.
Yes.
It was a colossus of an idea, though.
The role of Rhodes in science, or the colossus of Rhodes in science. Yeah, apobec 3. So, because obviously apobec 3 proteins played such a decisive role in limiting HIV-1 tropism in macaques, we thought we would continue our studies to characterize them. So there's very little that is known about the apobec 3 polymorphism in macaques. Usually they sequence one gene from a cell line and say, yeah, this is it. Yeah. But it turns out that's not the case. So that was a project that we started after we saw that we could get HIV in monkeys. It was to sequence the apobec repertoire of our monkeys and see what's going on there. And what we found is that the apobec 3 locus is extremely polymorphic in macaques. It's
More so than humans.
a lot more. So there's multiple variants of apobec 3F, multiple variants of apobec 3G, and apobec 3H is the most polymorphic. So from 14 animals, we recovered 13 different variants. And each variant has a splice variant too. So each monkey has 4 different ApoBec3H proteins going around.
These are from lab monkeys that have been bred in captivity, I presume, right?
Yes, but they're not inbred animals.
Yeah,
and we don't get them from, at the time, we didn't get them from one facility. So it's not,
Got it.
it's, I think it's, well.
It might be even more extensive if
It might be even more expensive
Yeah.
if you go to the wild and look at them,
right.
yes. So we're surprised to get so many proteins. And then we asked, so ApoBec3H is also polymorphic in humans. So it's the only one that has a high degree of polymorphism with both active and inactive alleles. And there's actually a geographical distribution of these alleles. And the active ones are more common in Africa. So, but it's nothing to the level of polymorphism that we see in pigtail macaques, at least. And we tested all these variants, and all the variants that we have tested are active to different degrees, but they all retain antiretroviral activity. And then they're all resistant to HIV-1 VIF. So
these are presumably because of SIVs that infect these animals, or?
Well, good question, because there's no known lentivirus infections in Asian monkeys like macaques.
Right.
SIV is pathogenic in them because it's an artificial inoculation. So presumably for some ancient infection by something, same as the trim sip that the pigtails have, it must have been selected. I mean, a protein that arose from retrotransposition must have been selected against a pathogen that was circulating. I don't know how many years ago, millions of years ago. So the other thing that we saw is that they all had, of course, they had amino acid changes as different variants. And at the time, Paul, well, still now, is part of a cRNA. That is a Center for the Studies of HIV RNA. And it involves multiple PIs that collaborate and worked in this particular center on the HIV-1 RNA. And he suggested, why don't you get in contact with Janet Smith and see if she would be interested in taking some of your variants and testing them for in vitro. Okay? So I got in touch with Janet, and she's found the project interesting. And we thought, okay, let's test the different variants. And the project really kicked off when Jennifer Bond, the first author, who is a graduate student, took over. She was really enthusiastic, and she really pushed it and started doing all these biochemical assays with the variants and setting up crystallization. And
I can fill in a whole lot with that because I met with her this morning. Whenever you're ready, but go ahead.
So it's we tested actually human ApoBec3H and 3 different pigtail ApoBec3H variants, and although they all behaved very as expected in the biochemical assays, only one pigtail ApoBec3H variant gave crystals.
And what were the biochemical assays that you were doing?
Deaminase assay and purification and yes.
Okay, got it.
So that was what's so amazing. And that differs from the other variants in perhaps 2 positions, but these 2 positions were apparently critical to get good quality crystals out of it. And I would say we would never have gotten the structure if we didn't work with the right variant. And that variant came about from very basic studies that someone might think, okay, they're not that interesting.
It's the role of Santorini.
Yes.
Why didn't you use cryo-EM?
Too small.
I don't know.
How big are these ApoBecs roughly?
They're very small proteins.
Because 30K,
yes.
According to the Nobel thing we read last time, 30K is like the lower
size limit
for cryo-EM.
That's the answer for that.
That's just
also, if you can get crystals, why do cryo-EM?
Ooh.
Yeah. Crystals, a crystal structure.
No, I hear you.
So it turns out that the ApoBecs are really hard to crystallize, as Theodora intimated. And when Jen got involved with it, she said she was trying all kinds of wild and crazy things to, well, first of all, to even get it purified enough, because the problem was it co-purifies with nucleic acid. And it took her 6 months to get to a homogeneous sample, which was a single picon size exclusion. But it still had RNA associated with it. The 260-280 ratio showed her that there was still nucleic acid there. But and this was after this extensive RNase treatment. But she set up trays anyway. And then it was this idea of Theodora's to try all the different isoforms, these different variants, and finding the one that binds less well to RNA. It has this, I thought it was a 1 amino acid difference in one isoform that enabled getting the crystals of the zeta isoform. Mm-hmm. But so that was really exciting in itself, because this was just after Jen had joined the lab. She had done a rotation in Janet's lab, but then chose a different lab for her PhD. And that was with Aaron Goldstrom. But then when he left to go to Minnesota, Jen wanted to stay at Michigan and had to go back to Janet, or went back to Janet, because she really wanted to work in Janet's lab. And she said that Janet asked her 3 separate times, "Are you sure you want to do this? Are you sure you want to do this?" And Jen was starting to get a little nervous. And she said yes for the 3rd time. And then Janet said, "I'd be delighted to have you join the lab."
Third time's a charm.
Yeah. So it was really exciting just to get this single peak by size exclusion, and then very exciting to get the crystals. And then it was basically only 2 weeks from when they finished the data collection to when they had the structure. But the really cool thing about getting the structure was, usually you do what's called molecular replacement, where you try and compare your molecule to other things that have already have a structure solved. But it doesn't work very well if there's a lot of scattering that's unaccounted for. And that was the case here. And so they used the fact that it binds zinc and did single anomalous diffraction. I just wrote down DIFF. I think that's what it stands for. To help them get the phase information. So they had lots of good amplitude data, but without the phase information, they couldn't get the structure. So she worked on that. She was basically solving her first structure, and she couldn't, she knew that there were 30 amino acids at the C terminus that were unaccounted for. And there was this region of density that she tried to fit these amino acids in, but it wasn't working very well. And there weren't enough aromatic amino acids to account for all the aromatic type structures that were in this unaccounted for density. And so the aha moment was when she said, "I think I found the RNA." So the amazing thing was that that's where the double-stranded RNA was. And so getting this structure was really exciting. And then that gave all kinds of information about how this gets into the virion and so forth that I'm sure Theodora can tell us more about. But when I was talking to Jen this morning, she was so excited about this whole project. It was almost like I had to pull her down off the ceiling because she's just really enthusiastic about it. But I could see that getting the purification and crystallizing it and then solving her very first structure. And there weren't people in the lab really who had done structures from what she said that were still in the lab who had done anything like it with a nucleic acid and protein. So she was really a pioneer in that.
Oh, that's great. Cool.
So tell us a little bit about this, Theodora, the fact that RNA was bound. Did you expect that to happen?
So expect that to happen.
Have RNA bound to the protein?
The RNA itself bound to the protein? No, that was not that surprising. It's what... As you just heard, it was extremely difficult to remove all RNA from the molecule. Extensive RNase treatment to the point where we only had 10 to 11 nucleotides of RNA left bound to each alphabet. What was surprising is to what RNA it's bound. It's bound to an RNA duplex. As far as we know, there's no other structure of a protein bound to an RNA like this. There's zero, it's a heterodimer, there's zero protein-protein contacts. It's all mediated via RNA.
Right.
So it's pretty amazing. And the actual duplex is very interesting because you have these 7 base pairs that form a duplex between the 2 molecules, and then a kink that's stabilized by the protein. And so that kind of gave us a clue that maybe this is physiologically relevant. Maybe this is what ApoBec 3H at least does, because it's different to the other ApoBecs. And what we used to look at that is an assay called CLIP-SIC. So it was developed actually by, specifically developed to look at viral protein RNA interactions in Paul's lab. So what it basically does is you radio label the cells, and then you cross-link your protein to the RNA. You extract the protein RNA complexes, and then you generate a library of your RNAs that you have bound to the protein, and essentially sequence everything, high throughput sequence. And then you can...
I just want to interject because the CLIP acronym always confuses me when I first see it, but it stands for cross-link IP. It has nothing to do with cutting or nuclease or anything like that. So just to help everybody else that might have that problem like I did.
Yes. Yes, exactly. That's what it is.
I don't know enough to have that problem.
Kind of like chip in a way, chromatin IP, except here you're using this protein bound to the RNA to bring it down. Yeah.
So we'd actually, I should say this, with a UV cross-linker that we bought off eBay.
You're kidding.
Whoa. Great. I've bought stuff for my lab off eBay too. It's
why did you get it on eBay? It was cheap?
Yeah.
Wow. It worked.
Buy one, get one free.
Well, I saw the other day on eBay, 90% of what they sell is brand new. So it's not all used.
Yes. So I forgot where I was now. The
CLIP.
So talking about the RNA that comes
the RNA, yes.
CLIP-SIC.
So then once you have your RNA sequence, what you could do, and it takes a lot of bioinformatics, it's a heavily involved process, you can map back everything to what your reference RNA is. And in this particular case, it's the HIV-1 genome. So we can map precisely where on the genome these ApoBec 3 proteins are bound with most frequency. And you get this graph that's in one of the figures of the paper, and you can clearly identify peaks. You can see that the binding is not homogeneous. The protein prefers certain regions of the genome to which it is bound. So I took the 10 most frequently bound regions and started analyzing them, thinking, maybe I will find some sequence similarity and then we'll solve, because the way that ApoBec 3 proteins get incorporated into particles is unknown. It's an extremely promiscuous method, yet it has to be specific because you have viral RNA constitutes a very small percentage of the total cellular RNA. So somehow the protein has to find the viral RNA and specifically interact with it, yet not so specific that it will only block one set of viruses. It has to be broad. So I looked for sequence specificity and there was nothing. I mean, it was just so disheartening. And then I thought, all right, let's look at other features. And I ran a M-fold and looked at the secondary structure of RNA. And there it was obvious. All the peaks had the potential to form this 7 nucleotide duplex.
Duplexes.
And then we took what we called valleys, so region where the ApoBec didn't bind so strongly, and analyzed those. And out of 10, only 2 had the potential to form the 7 nucleotide duplex. So clearly there was a very obvious difference between the potential to form secondary structure with 7 nucleotide base pairs in the regions where ApoBec 3H bound versus regions that it didn't like.
How many molecules are bound to an RNA? Do we know? A viral RNA?
No.
We don't know. Go ahead, Alan.
So this type of duplexing is a feature that is probably distinct to or primarily found in viral RNAs, I'm going to guess.
Well, that's what we hypothesis of is that if the viral RNA has a propensity to form these 7 base pair duplexes, it has to be 7. It could be longer, but at the minimum has to be 7 based on the structure. Has a propensity to form these structures more readily than cellular RNA, then this would be a mechanism by which the ApoBec3H proteins would specifically recognize viral RNA and then ignore the rest of the cellular RNA.
Mm-hmm.
And correct me if I'm wrong on this, but isn't viral RNA somewhat more likely to be folding up in the cell because it's encapsulating, whereas cellular RNA is more likely to be unfolded and being translated?
I don't know about that.
Or is that
okay? Certain regions of the viral RNA, certainly. So for ApoBec3H, the most preferred binding site is the 5' leader sequence of HIV1. And that is very well studied. And it's known that it forms secondary structures. It's known also that the primer binding site is there. So there you have a duplex already. So it makes sense that it likes to bind there. And this could actually also account for the ability of ApoBec3H to block virus infectivity by deaminase independent mechanisms. So you can imagine that if it's docked onto the leader sequence, then it will inhibit reverse transcription from happening.
And I guess it would also be, I mean, there'd be a strong selective pressure once you have a system like this in the cell not to evolve RNA sequences that are going to do this.
In cellular RNA.
In cellular RNA.
Yeah.
Yes.
So then you'd have the ApoBec as a system that can essentially recognize something as not being from around here, and therefore potentially a threat, right?
Perhaps. Yes, perhaps.
Okay.
So can you? The reason I asked you how many binding sites there would be in the HIV genome is, could you change all of them so that ApoBec no longer binds? Would that be possible?
We'd have to. Yeah, we'd have to do that. The problem. Yeah, this.
Then it would be Then it would be VIF independent. You wouldn't need VIF to antagonize ApoBec. This one anyway.
So this,
I'll bet you that messing around with that 5'
I bet
M guy is going to cause problems.
I would bet the virus has already done this experiment.
Well, that's the next question. Why? For what reason does the virus have VIF instead of simply changing all of these duplexes and avoiding ApoBec binding, right? We don't know the answer, but
but the virus has had a long time to solve this problem.
Yes.
And the fact that it chose to do it with VIF tells you that it's unlikely that you'll be able to change everything.
Still, it would be interesting to try it, right?
How many molecules of ApoBec are typically packaged in a virion? Do you know?
No, I do not know this answer. ApoBec3H, actually, incorporation compared to other ApoBecs, at least in our hands, from what we can say, is lower than other ApoBec3 proteins. However, I've never seen these experiments done with physiological levels of expression of ApoBec3 and estimate exactly how many molecules are in there. I mean, maybe someone has done it, but at the moment I haven't seen it.
I mean, that's an important question, because if there are only a few binding sites and a few molecules in a virion, it would be an easy experiment to change. Maybe it just takes one base change to disrupt the duplex, and you could conserve protein coding and all that if you're in a coding region.
I'd probably be able to do this for other sites, but not for the primer.
Yeah.
For the 5' leader, as someone mentioned in the panels, but it will be very difficult because that performs so many functions that it's.
So what's interesting here is that the original RNA that is in the crystal structure came from E. coli.
Yes.
So maybe our listeners don't appreciate that, but it's pretty nonspecific binding. As long as it's at least a 7 nucleotide duplex, it will go in there, sequence independent. In fact, I think you say that the structure showed that at every position, you couldn't tell what base it was.
No,
because there are all sorts of other sequences in there.
It's promiscuous. So the peaks are slightly G-rich because G has the ability to pair both with T and with the U in RNA. So that would make it more prone to make this kind of structures. But other than that, there is no sequence specificity. And the same, the different RNA molecules do not have a sequence.
So I have some questions about the fact that it shows up as a dimer, okay? Which it appears as a dimer, and yet the proteins, it's a dimer because of the interaction with RNA. The proteins do not interact with each other.
Exactly, yes.
Okay? So when when people try and purify it and they get a dimer, it's not because of a protein-protein interaction. It's because the RNA is locked in tight.
Yes.
Right? So what interests me about that is that somehow, if it's showing up primarily as a dimer, that suggests to me that the binding of, if they're not binding, the proteins aren't binding to each other, suggests to me that the binding of one protein to the RNA promotes the binding of a second protein. Otherwise, you'd have a bunch of monomers lying around. Okay? So that it suggests to me that there's some sort of cooperativity that goes on that favors this dimerization.
Yeah.
Does that make any sense?
Yes.
And so I'm thinking, that maybe binding one tweaks the RNA in a fashion that, enhances binding of the second guy. That's very interesting.
If you could get this thing purified apart from RNA, you could actually do that experiment biochemically, couldn't you?
Right. And I was trying to figure out a way to do that.
But the first part of that is the hard part, is getting it purified apart from the RNA.
Well, you could probably synthesize this protein, APROBEC, right?
Right.
But again, as we said, we did, yes. So we did try to remove RNA very, very hard, and it proved impossible.
Right.
I was wondering if you could even chemically, if you could do some sort of mild base treatment over time or something like that, without completely trashing a protein. You might have to refold it. I don't know if there are peptides that are sensitive to that kind of treatment or whether you get away with that. I don't know. I was trying to think about that.
I have to talk to Jen and Janet about this.
Yeah, I wanted to point out, because we haven't mentioned it so far, but the duplex has the two 5' ends of the RNAs pretty near each other, but that kinking then allows the base pairing to occur. So there's 2 nucleotides that are not base paired and then 7 that are. And also that this is A form structure nucleic acid. In this case, it's RNA, and that is evidently also the form that RNA-DNA duplexes tend to take. So there's some speculation, at least on Janet's part, and I don't know how Theodora feels about it too, but that maybe the ApoBec3H is binding to the RNA-DNA heteroduplex. That's the first product of reverse transcriptase.
Yes, actually, thank you. I was trying to find a way to bring this in. So we know that ApoBec3 proteins are active during reverse transcription. And we also know that the reverse transcriptase moves much faster than the RNase H. So the target is the single-stranded DNA. So the RNA has to be removed, but you could potentially have these short-lived heteroduplexes formed because RNase H is slower that would also form this A-form duplex that would potentially feed the DNA into the active side of the molecule. So there could be also a way that ApoBec3H deamination is promoted. So yes, that is also a possibility.
So do you think this is the mechanism of bringing ApoBec into the virion?
I like that interpretation better. I mean, one does not exclude the other. I suppose could work, but that would provide a very, for me, elegant way of getting into the particles without being dependent on sequence.
Right. And would this be, you think, universal for ApoBec incorporation?
No, I don't think so. I think the ApoBec3G structure and 3F have been solved, but only individual domains. So and they are quite different. So the N-terminal domain of ApoBec3G, that is the one that binds RNA but does not deaminate, is already heterodimer. It's a protein dimer. Right. And the RNA binding region is created by the dimerization of the 2 units. It's not as basic as ApoBec3H. So there must be another. And it also doesn't like the same regions as ApoBec3H. It doesn't have this preference for 7 nucleotide duplexes in the RNA.
Right. There's always more than one. Someone said that recently. There's always more than one way of doing things. In biology, anyway. That's a great story. Love it.
Thanks.
Beautiful. And I like the serendipity or the Santorini aspect. That's going to be a new phrase for serendipity, the Santorini effect.
Santorini's only half of an island, though. Remember that.
Yes.
Yeah, okay.
I wanted to tell a little bit more about Jen. So she changed labs in the middle of her third year, and she's recently gotten her own individual F31 award to continue this fellowship. But she started out as an undergraduate in Mike Summers' lab at the University of Maryland, Baltimore County, where she worked on 5' leader dimerization mechanisms for SIV. And so she's always been really fascinated with RNA. And so when she came back to Janet's lab, that was something that she really wanted to do. And Janet said, that will be fine as long as you also do protein biochemistry. So I thought that was a kind of an interesting twist. And although she's got this fellowship and so on, she is thinking somewhere down the line of an academic type of career. So I think was really exciting to find out too. She really likes mentoring students. She's had 2 undergraduates, one just recently started and one that she's been working with for more than a year, I think she said. So it was really fun to, like I said, talk to her and just get excited by how excited she was about all the structural aspects of it and all the biological aspects of it.
Theodora, can you give us a sense of where you're going with some of this work?
So let me first say that, yes, working with Jen has been great, and Janet. And Jen is extremely enthusiastic, and she's very receptive. So whenever we talk about virology, she really listens, and then she's just so eager. It's amazing to work with people like this. So where are we going with this? Yeah, we're trying to do several things to verify how much of our predictions are actually correct. So, for example, what will happen if we mutate residues in ApoBec that are shown by the structure to be important for RNA binding? I suspect those studies are going to be difficult because so far the mutants are either not well expressed or if you mutate, the basic nature of the protein makes it difficult to mutate. And then also probe a little bit more the interaction with VIF based on the structure and do some mutagenesis studies with that. So, yeah, that's where at least this project is going to go.
I think it would be cool to try and get ApoBec into a different virus.
Oh, it's a good...
Moving one of these little 7 or more duplex sequence. Somehow, it would have to be a virus with a DNA phase, right? And you'd have to somehow bring it in via RNA. But even a different retrovirus that normally doesn't deal with ApoBecs, right? There are plenty of those, right? I mean, the Maloney doesn't have ApoBec, does it?
Yeah, no, Maloney is restricted by ApoBec.
Are any not?
I don't know of any that are not. So you would have to move to a different virus.
Maybe foamy viruses are not.
Oh, no, foamy actually have a different protein that counteracts ApoBec 3 proteins. Yeah. So
maybe, so Hep B has a reverse transcriptase.
Maybe, yeah, that's what I was thinking.
So that could be, anyway, that would be cool if you could get it to target a different genome.
Yeah.
How about Trichinella, Dickson?
Why not?
All right, if no one has any further questions, let's move to some picks of the week. Do you know what picks of the week are in TWiV? I don't know if you have one, but if not, we can let you think while we're doing ours. Okay. And Dickson, what have you for us this week?
Well, I have a visual, and I picked this one specifically for Kathy because I think you like to watch things move in color. So it's a video of pouring paints over inanimate objects and watching the flow rates and how the sizes and shapes of things change. I just got fascinated with the structure of this thing, and I wasn't particularly interested in how you did it or why you did it. I was just interested in the artistic aspect to it. And if you go to the website, you can actually see these wonderful, it almost looks like a moving art project. It actually moves with remarkable hypnotic colors.
They misspelled sheer.
Don't look at me. I didn't.
The sheer joy of watching paint.
No, I think they wrote that on purpose.
Really?
Yes.
Yeah, I think so.
Because the sheer forces in the paint, maybe?
Exactly right. Exactly.
Dickson, do you remember those things at fairs where you would go and paint on spinning cardboard? It just reminds me of that.
A little bit. I mean, maybe.
It's really cool.
But
There was a home version of that, too.
Yeah,
it was,
there was.
a spin art?
Yeah.
Spin art.
Yeah.
Exactly.
Yeah, you could set up a little turntable and then you drip paint on it and it's spun.
Sure. So I actually once made an abstract painting using this technology.
I don't know. I think many of your paintings are abstract. Like
some of them are, yeah. And I got some automobile undercoating.
Yeah.
And I sprayed it on a piece of masonite, a big piece about the size of this round table that we're sitting at. And I let that dry. And then I took car enamels, automobile enamels that you can buy. And then I started to pour them onto this large structure of various colors. And sometimes the colors would go underneath and come back up again in different places. And then I'll let the whole thing dry. And it was quite amazing, actually, because it's all by accident. But I think it's due to the physical properties of each of these paints. And you can see they're not mixing. They're remaining separate, but they form patterns. And the patterns have something to do with their sheer force quality.
Dickson, is it art?
I think it is, actually. Yeah, I think it is. Okay. And very often there's a lot of art in nature.
Oh, yes.
And science.
Science, too.
Oh, there's no difference, I think. And you're at Rockefeller now. You can see that the art all over the place just absolutely speaks to that issue. Very inspiring.
Nice. Thank you, Dickson.
You're welcome.
Alan, what do you have for us?
I have a time-lapse video. This was shot from the bridge of a cargo ship, and it's 30 days at sea. And the guy did a really good job steadying the camera. So you get to just watch this ship moving across the ocean through day and night, and beautiful views of the sky and the weather systems blowing through. And it's just mesmerizing. You can watch it with the sound on. It's got some music to it, or you can watch it with the sound off, and it's just really. And he's got subtitles saying where this thing is going. Cool. And this, of course, is the way most of the world's stuff moves around. It's on these huge container ships out on the ocean, and it gets out on the ocean, and it's just this tiny little thing, and there's this huge sky above it, and just a real fun video.
I guess he just fixed the camera on the bridge somehow, right?
Yeah, it was it was bolted on, but I think he also probably did some kind of trimming to the video because you don't really see a whole lot of rocking.
It's probably a GoPro, I would think.
I assume so. Yeah,
it's neat.
I was disappointed you didn't go through the Panama Canal. So
right.
So sure enough, I looked up on YouTube, and you can find a time lapse of going through the Panama Canal. Okay,
yes,
that's pretty good too.
Yeah.
Rich Condit, what do you have for us?
Well, I went to my library. I'm amazed this hasn't been picked before.
Yeah.
I'm recommending Hitchhiker's Guide to the Galaxy by Douglas Adams. This is in the really way out cult science fiction / humor category.
This is pretty mainstream, I think.
Yeah.
Don't watch the movie. Read the book. And Kathy, you have a note here. You've listened to the audiobook?
Yeah. A long time ago, more than 20 years ago. It's really good.
Well, this was wasn't this a BBC radio show?
Yeah, that's what it came from. Yeah.
And if you're not familiar with this and you read the book, you will all of a sudden be enlightened about a number of different memes that go around, like goodbye and thanks for all the fish.
And 42.
Yeah, 42. Which is the name of a episode of ours.
I left that on my wall of the lab I moved out of.
42?
Yeah.
That's good.
So my favorite, I think, is Sorry for the Inconvenience.
Yes.
That's right. Exactly.
Sorry for the inconvenience. Great. Kathy, what do you have?
Well, I picked something that's, at first it sounds like it's going to be kind of silly. What happens when you have 25 million Orbeez in a swimming pool? Do you sink or float? And so if you haven't watched this yet, you could think about it. And I, first of all, didn't know what Orbeez were, but they're some activity toy that you can buy. They start off as tiny beads, and then they grow to 150 times their size in water. And so these guys fill a whole swimming pool with these Orbeez, and the guy the main guy who's doing this is called the backyard scientist. He makes a prediction of whether he himself will sink or float, and then you can see what his result is. And then he talks about the science behind it. And so it eventually comes around to that. And then he invites over some kids, and then you see them playing in this pool full of Orbeez. But I thought it was a nice mixture of science and fun.
I couldn't stop thinking, how is he going to clean all these things?
Yeah. Rich, I think this is Florida. He's got a screen over his pool.
He does. I noticed that. Yeah, it looks like Florida.
Screen cover, which I would like to have now because the leaves are getting ridiculous. That's pretty cool.
You're presuming he actually would want to remove these.
Right.
This actually looks like a lot of fun.
Well, it would keep the heat in very well, too, right?
Yeah.
So you don't have to heat it as much.
The best part of the video was throwing molten aluminum in and pulling it out because see what weird shapes it would form.
Yeah.
Neat. All right, I have two picks. One is the latest episode of This Week in Evolution, TWiV. Our guest, Nels and our guest was Marco Vignuzzi. Does anyone know Marco? No?
Yeah.
He is a evolutionary virologist at the Pasteur Institute, happened to be visiting Nels. And they just did a cool piece of work in his lab where, so he works on RNA viruses that exist in a sequence space, right, as a swarm of mutants. And he asks the question, what happens if you push it to a different sequence space? Is that a problem for it getting to where it needs to go? So the way he changed the sequence space of Coxsackie virus and influenza A virus, he changed all the serine and leucine codons so it would only require one mutation to make a stop codon. And then these viruses replicate fine, but if you mutagenize them, they lose fitness. Or if you passage them or put them in animals, they are not fit because you've put them what he calls in a bad sequence neighborhood. So it was a lot of fun. He's great to talk to. The name of the episode is Good Viruses Visiting Bad Neighborhoods. And this is worthy of an Alan Dove title. Yes. Don't you think?
That's good. I like it.
And Nels is
my seal of approval.
Nels is actually pretty good at it. I let him do the picking. And then I have one small pick, which is a website called Every Time Zone.
This is neat.
If you need to know what time it is somewhere, this will tell you. It has a nice green line, which is the current time, and then you could see everywhere else, well, in main places of the world where you're likely to be near, right? Honolulu, San Francisco, Denver, Chicago, New York, London, Vienna, Mumbai, Singapore, Tokyo, Sydney, Auckland. It would be nice if you could add your own, but I think it's pretty great because I don't know if do the right thing. I always miscalculate when I try to figure out the time zones. And I think there are similar apps that you can get for your phone as well. So Theodora, you have anything? Oh, you got one. That's called World Clock. Yeah.
Yes.
Yeah, that's an app on your iPhone. That's right.
Nice.
What cities do you have on that?
New York, Athens, Melbourne, London.
Right.
So New York, Athens, I can understand.
Melbourne is actually my friend Gilda Testian.
Yeah.
She was a postdoc here with Steve.
Yeah.
So occasionally we call each other, so I have
You
to
wanna know
make
the
sure
time.
I don't call her in the middle of the night.
I visited her in Melbourne. We went to dinner.
Yes,
Lovely.
that's great.
I was at a meeting and we went. And she was on a TWiV, actually, from Melbourne.
Yes.
They told me that's how you pronounce it, Melbourne.
Spent 6 months there, and I can guarantee you that is exactly how they pronounce it.
So I
Melbourne.
I gave a talk, and I got up there, and I said, thank you for inviting me, Melbourne, and they all applauded. Then I said, Geelong, which is a neighbor, and they applauded again. It was great.
Not Geelong?
Geelong. No, do you have a pick for us, Theodora?
I didn't think of this in advance. I am currently reading To Kill a Mockingbird.
Oh, yeah.
Lovely.
Because I thought that I did not have, I know it's a classic staple in American
Sure is.
Literature and what kids read, but since I didn't grow up here, I've never read it. So I thought I read it. Yeah. So
kids read it, they don't understand it. Adults read it, they understand it.
That's great. Yeah. Have you read Aerosmith?
No, I haven't.
Should read it.
Yes.
Another good one.
What is your favorite ancient Greek play?
Oh, what a good question. Are there a lot of them?
What's your favorite Greek playwright?
I prefer the comedies. I prefer Aristophanes to the tragedy. Yes, and I think he's so. Sometimes it's so subtle that I don't think people get it. How critical he was of politics and politicians. So I like
frogs or
the birds. I don't know how to say the names in English actually because I know them in Greek. Is one or Lysistrati. Lysistrata. It's very, very good.
Sure.
That's one of my all-time favorites.
Remember those from college?
Absolutely.
Yeah.
If only we could employ those technologies to stop the wars.
Can you say something? Can you say something in Greek for us?
It's,
Please. Wow.
No idea.
Beware of the Greeks that bring gifts.
Great.
I think a Persian advisor said that to the king.
There's an infection called Echinococcus granulosus, and it's a very large space-filling lesion. And one of our teachers used to say, and it was very popular in Greece because that's where they raise a lot of sheep, and there's a lot of sheepdogs, and a lot of sheep raising people like shepherds get this infection. So when they come to this country, they said, beware of Greeks bearing cysts.
Have that.
And he got a big laugh, and then they have to understand why that's a joke, because it's not such a joke, obviously.
Yeah, cool. All right, we have a listener pick from Fernando. He writes, a story arc completed. And this is Lego is finally celebrating the women of NASA with their very own set. This is very cool. Lovely. So we have Margaret Hamilton, Nancy Roman, Mae Jameson, and Sally Ride.
Nice.
Who you know have all played important roles. And it's a lovely Lego with a shuttle, telescope,
nice
books, computer discs, maybe.
I think that's manuals for. Wasn't Margaret Hamilton the Apollo computer?
Yeah, she was. She developed a lot of the software for the Apollo launch.
So Wright and Jemison were astronauts.
Right.
Hamilton was this computer scientist who built the programming used in Apollo missions.
Yeah.
Roman was one of the leading astronomers behind the Hubble telescope.
Wow.
It's a beautiful Lego. Just
231-piece set.
Nice.
Very nice.
Beautiful. It would go right nice next to the scientists, right? What is that called? The women scientists in the lab? There's a name.
Something like that, yeah.
I just bought Amy one on eBay for getting her paper accepted.
Oh, nice.
Oh, it's looking. Oh, eBay. I should look at eBay.
eBay has them. If they're out of production, it's easy to get them on eBay. They're not too expensive. But I said, don't open it until you have your own lab. Because that's when you want to build it, right? Yes.
I just saw this thing that the set was supposed to include this astronaut, whatever set, was supposed to include Katherine Johnson, the mathematician whose calculations made Mercury and Apollo missions possible. Yeah. It's one of the subjects of the movie Hidden Figures, but her family declined to allow Lego to use her likeness. So they tried.
That's interesting.
Right.
That's interesting, because I was kind of looking for her,
yeah, I was too.
It's too bad.
Yeah.
But it'd be so cool.
Yeah, I would like to be a Lego figure.
As Dickson would say, it's a national treasure, right?
Yes, treasure.
Why would you want? You have to recognize when you're a national treasure. You have to be in the public eye, right, Dickson?
Yeah.
Yep. All right, that is TWiV 465. You can find it at Apple Podcasts, microbe.tv/TWiV. And you can also find it on your favorite podcast app, which you can find on your cell phone or on tablets, even laptops and other kinds of computers. And I would just like you to subscribe to TWiV and TWiP, sure.
All of them.
Just subscribe, and you will get every episode. I know some weeks you can't listen, but subscribe, and it gets downloaded automatically. That will help us with our numbers, and we'd appreciate it. If you like what we do, you can support us financially. Go to microbe.tv/contribute for the ways you can do that. Dickson Despommier is at thelivingriver.org, parasiteswithoutborders.com. verticalfarm.com. He just had a new blog post.
I did.
Amazing.
I did. I squeezed it out.
Thank you, Dickson.
You're welcome, Vincent.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
Alan Dove is at turbidplaque.com. He's also on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiV and the sponsor of this episode, the Defense Threat Reduction Agency, and of course, Ronald Jenkins for the introductory music. You can find him at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
Φοβού τους Δαναούς δώρα φέροντος.